So one thing we haven't talked about yet is where all of these metabolic processes take place. And for aerobic respiration, the mitochondria is really important. And so I'm going to introduce you to the mitochondria. Let's just type it out here. So this is not a cell that I've drawn here. This is the mitochondria. Mitochondria. Okay. Maybe I can make this a little bigger. There. This is, free internet software. So, all right. So I want to label the different parts of the mitochondria for you. So let's take a look at it. All right. So we talked about the mitochondria in last week's lectures, right? On eukaryotic cells. Mitochondria are an organelle inside eukaryotic cells, and we talked about them a lot. They are energy processing centers for the cell, and we talked about them in regards to endosymbiosis theory. So mitochondria and chloroplasts, when it comes to plants, were once bacteria that got endocytosed into a very ancient, larger fermenting cell, and started living symbiotically together. And the mitochondria, these little organelles, these little bacteria, gave the larger cell this power of aerobic respiration, which is this really efficient way of extracting energy from food. So when we talked about endosymbiosis theory, I mentioned that the mitochondria have two phospholipid bilayers, two cell membranes, right? And this is one of them. So this is a phospholipid bilayer, just like the cell has. And we call it the outer membrane, right? So let's type that here. I want to reduce the size again. Oh. Outer membrane. I'm not sure what happened here. Okay, outer membrane, right? I can't move it. Okay, so this outer gray line is the outer membrane, the outer mitochondrial membrane. That's easy to remember. And you might have guessed that the inner gray line right there is called the inner mitochondrial membrane. And that's the wavy one. It's very, very wavy. And it's wavy because that increases its surface area. So this inner membrane can actually be longer, but squished into a smaller space by being wavy. And there wants to be a lot of area here because there's more space for enzymes involved in the electron transport chain. Then between these two membranes, we have what's called the intermembrane space. So if you go, there's one membrane, there's another membrane. The space in between them, which is narrow in some places and wide in other places due to that wavy nature of the inner membrane, we call this the intermembrane space. Intermembrane space. Inter means between. So if you hear the word interspecies, that means between species. If you hear the word intermembrane, it means between membranes. So that's the intermembrane space. And then the last part that's important is this region that's in the middle of all the membranes, just in the very center, is called the mitochondrial matrix. All right. So where are all of the different metabolic processes happening? Well, let's take a look. I'm going to take another color here. Okay. Glycolysis does not happen in the mitochondria at all. It happens in the cytosol of the cell. Remember, the cytosol is just the liquid inside the cell. So glycolysis doesn't happen in any organelle. All the enzymes that drive all the different steps of glycolysis, they're just floating around willy-nilly in the cytosol of the cell, just free floating, okay? Now, remember, in glycolysis, glucose goes in and two pyruvates come out. Okay, and then the pyruvate goes into the mitochondrial matrix. Oh, the matrix turned blue. That's okay. So the inner mitochondrial matrix, that's where the pyruvate go, and that's where the citric acid cycle happens, or the Krebs cycle, whatever you want to call it. So Krebs cycle, or the citric acid cycle, happens in the mitochondrial matrix. And I wish I could move this, but apparently I cannot. Oh, there we go. Okay. Okay. And then the last piece, of course, is the electron transport chain, which I'll abbreviate ETC. That happens in the inner mitochondrial membrane. Okay. All right, so you have glucose brought into the cell and it's just floating around in the cell cytosol all around. I don't have the cell drawn here. I just have its tiny little one tiny little mitochondria drawn here. There's many mitochondria in each eukaryotic cell. So in the eukaryotic cell, there's glucose floating around the cytosol. Glycolysis is happening, breaking it down into pyruvate. The pyruvate gets shuttled into the mitochondrial matrix where the citric acid cycle happens, and that's where all of those high energy electrons are stripped. Right? And then those high energy electrons are taken to little tiny enzymes that are embedded in the inner membrane. Okay, and so the enzymes, the enzyme complexes of the electron transport chain are all anchored here in the inner mitochondrial membrane. Okay, but the intermembrane space is also really important in the electron transport chain. So I am actually going to zoom in on a section like this. So we're going to zoom in here. We're going to get a section of each membrane and the intermembrane space. And in the next slide, I'm going to clear this board, this whiteboard here, right? I'm going to clear it, and we're going to just zoom in on this and expand it out to take up the whole screen of the next board. So here we are zoomed in on the inner and outer membranes of the mitochondria. So inner membrane, outer membrane, here's the intermembrane space, or the between space, and the mitochondrial matrix. So I like to draw this piece because I think it's just easier to follow that way. So let's see here. What is the best? You know what? I'm just going to use this, the larger size. Okay, so first we have NADH and FADH2 coming from the matrix, right? Because we said that the citric acid cycle happened in the matrix. So we have NADH down here at the bottom, and we have FADH2. Right? This should be a subscript. I don't think that I have that capacity with this. Anyway, so they come and they pass their electrons, their high energy electrons that they stripped from food molecules, to enzyme complexes that span the whole membrane. And there are 4 enzyme complexes. There's enzyme complex 1, which is a Roman numeral 1. There's enzyme complex 2. And I'm drawing enzyme complex 2 small on purpose. Okay. So enzyme complex 2, enzyme complex 3. 1, 2, 3. And enzyme complex 4. They have other more complicated names, but it's perfectly accurate to call them. Oops, actually, you know what? I'm going to move this over a bit. Perfectly accurate to call them enzyme complexes 1, 2, 3, and 4. All right. Now, NADH is going to pass its electrons to enzyme complex 1. And that means it reduces enzyme complex 1 and it becomes oxidized. FADH2 is going to pass its electrons to enzyme complex 2. That means it becomes oxidized, it reduces enzyme complex 2. Now they're back into their electron poor form as NAD plus in this case, and FAD. Right? And the electron poor forms are going to go back into the matrix and then strip more electrons off of more food molecules, right? So they just get recycled, going back and forth between the NAD plus, strips electrons, becomes NADH, comes and passes them to the electron transport chain, becomes NAD plus, goes back, strips electrons, becomes NADH, back to the electron transport chain, drops off the electrons, becomes NAD plus again, right? Same thing with FADH2 and FAD. So let's start with the electrons that come from NAD+. I actually want to get this little star-like thing for the electrons. All right, so the electron is now here, okay? There's the electrons. So they're going to get picked up by a shuttle, an electron shuttle, and we're going to call it Q. Q for ubiquinone, but it's Q for short. So Q equals ubiquinone. Ubiquinone has the root of the word ubiquitous in it. Ubiquitous means found everywhere. Q is it's found in a lot of places in a lot of living things in nature, often in electron transport chains, but sometimes in other places. So Q is really ubiquitous. It's found across many forms of life. Okay. So these electrons now are going to get passed to ubiquinone. That means that ubiquinone oxidizes or takes electrons from enzyme complex 1 and it becomes reduced. So here's these electrons here. Then ubiquinone goes ahead and, now when that happens, I should say before we move on, when that happens, I want to get another color here to keep things clear. When electrons move to ubiquinone, they lose some energy and they gain some stability. So they've lost energy, and that energy has to go somewhere, of course. So what happens, what that energy gets used for is this enzyme complex 1 is actually a proton pump. Protons are the same thing as H plus ions. I'm just going to draw these instead of text writing them. So the energy released from that electron when it was moved is used to pump protons or hydronium ions from the matrix into the intermembrane space. Okay, so we've pushed some protons here. All right, so let's leave that at that for now. Now, ubiquinone can move through this membrane back and forth, okay? It's a shuttle. So it can move through this sea of lipids back and forth between the different enzyme complexes. So it is now, if I can go back to blue, probably won't be the exact same color, sadly. It is now going to go reduce enzyme complex 3, meaning it drops electrons on enzyme complex 3. So here are these electrons now. And then there's another electron shuttle that acts much the same way as ubiquinone, and it's called cytochrome C. I'll call it Site C for short. So it just floats through this membrane like a ship on a sea. So that's cytochrome C, Site C. And I'll put a little text box for it here. Site C equals cytochrome C. Okay. So now these electrons are going to get passed to cytochrome C. And that means that cytochrome C has oxidized enzyme complex 3 and it's become reduced. So we're going to go back to red. When these electrons are passed to cytochrome C, when they leave the enzyme complex, they become a little more stable, which means they release some energy. That energy, again, doesn't just disappear. Energy doesn't just disappear. It's used to do something. So it's going to be used. This enzyme complex is also a proton pump. It's going to pump protons from the matrix into the intermembrane space. I say proton when I'm talking about hydronium ions or hydrogen ions because hydrogen has one proton in the center and that's it. So if they don't have any electrons orbiting, it's just a proton by itself, right? So it's the same thing as hydrogen with a positive charge with no electrons. All right, so those hydrogens, again, are getting pumped from the matrix into the intermembrane space. So what you can see that we're doing here is we're transferring the energy that was in the electrons to a chemical gradient. It's actually an electrochemical gradient because there's positive charges. That's an electromagnetic charge. So you get positive charges built up on this side of the membrane and no positive charge on this side of the membrane. You also get a lot of hydrogens building up, a high concentration of hydrogens building up here, and fewer over here. So you have this gradient of high hydronium ion concentration, high positive charge, to less of those things on this side of the membrane. And that's unstable. You know, when we were talking about thermodynamics and energy and potential energy, okay, well, this is that example, right? When you have a gradient that's, you know, different concentrations on different sides of a membrane, that's unstable because it's not random. these hydrogen ions are no longer randomly distributed. They're being stuffed here, and there's not any over here, okay? So that's not a random distribution, that's compartmentalized, that's organized, right? It's like stuffing all the socks in your sock drawer instead of having them all spread out through your room. Random distribution of socks is when they're all over your room, equal to, equally in the sock drawer as out. But if you compartmentalize things, then all the socks are inside the drawer, inside a compartment, right? So here you're putting all the hydrogen ions inside a compartment, and that's unstable. So first you had um, potential energy in the food, stored in the chemical bonds of food. You released that and passed that energy to NADH when you put the high energy electrons in NADH. Now we're putting the energy into a proton gradient. Okay, and then that proton gradient is going to be used to generate ATP, as you'll see later. So let's finish this up here. So we've got to go back to blue. So cytochrome C can move through the enzyme like a boat through an ocean. Oh, there we go, like that, right? Okay, and it moves those electrons to enzyme complex 4. And when the electrons move to enzyme complex 4, they become even more stable and even lower energy, right? And then at enzyme complex 4, they are so stable at this point. They've been moving to, the only reason they've been able to move spontaneously is because they're moving down their energy gradient. So every time they move, they release some energy and they become more stable. At this point, these electrons are so stable where they are in enzyme complex 4 that almost nothing can remove them. The only thing that can remove them is atmospheric oxygen. So oxygen comes, actually I'm going to keep it in blue because we're talking about the electrons here. Atmospheric oxygen comes. This is the oxygen you breathe. So you've breathed it in, it's gone into your bloodstream, and your bloodstream is taking it to all the cells, which pass it to the mitochondria. And what that oxygen does is it takes those electrons. When it does, it gets split into H2O. So it picks up some hydrogens with those electrons. So that when the oxygen picks up those electrons, it gets split, picks up some hydrogens from the surrounding solution, and becomes water, H2O. Okay, one oxygen molecule will become 2 water molecules. And that's because these electrons were passed to it, right? So now those electrons are here. And I'll draw this line showing where they go. Okay. All right, so this is the only reason you breathe oxygen. The only reason. It's the only positive role oxygen plays in your body. Actually, and we'll talk about this later in the course, but oxygen is toxic to you. It wreaks havoc on your cells. It is this incredibly good oxidizer, meaning it just steals electrons from things, and that really disrupts a lot of molecular bonds. So it is not that great in a lot of ways, but we need it to clear the electron transport chain. And we'll talk about that in a moment, but I want to finish up and remind you that this is also a proton pump, enzyme complex 4. So when those electrons get pumped, when those electrons move to one more time to oxygen, they release some energy, and that energy is used to pump protons one last time into the intermembrane space, transferring that energy to this electrochemical gradient right there. All right, so here we go. All right, so if you didn't have oxygen, you would suffocate. And what does that mean? What does it mean to suffocate? Well, it means that this whole process shuts down. Okay, so if you didn't have oxygen, the electron transport chain would shut down because these spent electrons could not be removed. And if they can't be removed, these electrons can't move there. They can't move anymore. And if these electrons can't move, then these ones can't move. And if these can't move, then these can't move. And basically the whole assembly line gets gunked up. It all grinds to a halt. If if electrons stop moving through the enzyme complexes, protons stop being pumped into the intermembrane space. If protons stop being pumped into the intermembrane space, ATP synthesis will halt, and the cell will not make enough ATP to live. ATP synthesis stops. That is the energy currency of the cell. So all the energy economy shuts down. All energy requiring reactions stop. The cells can't move, they can't function, they can't move things across their membrane, they can't grow, they can't repair, they can't do any of the life-giving they can't do any of the biological life-giving reactions that they need to do without ATP. And so the cells die in a very short amount of time. They burn through the ATP that they have in a very short amount of time, and then they die. That's what suffocation is. So if you stop breathing in oxygen, you will not generate ATP. So I know we haven't finished this yet. You now are probably wondering, how does the energy in this proton gradient drive the production of ATP? And we'll get to that in the next slide. I just really wanted to drive that home. And another thing I want to drive, so without oxygen, electron transport chain grinds to a halt. No chemical gradient is generated. And without the electrochemical gradient, no ATP synthesis can happen. And so ATP synthesis stops, cell doesn't have any energy anymore, and it dies. So one last thing to say about this is we haven't talked about enzyme complex 2 and the electrons coming from FADH2, have we? So let's move to that next. All right, we have electrons from FADH2 are passed to enzyme complex 2. Oops. These go to ubiquinone, and then they follow the same path from there. From ubiquinone, they go to 3, to cytochrome C, to enzyme complex 4. The only difference here is that the reason I drew enzyme complex 2 so small is that it is not a proton pump. So protons are not pumped when electrons move from enzyme complex 2 to ubiquinone. Okay. And that means that the electrons coming from FADH2 do not contribute as much to the energy in this electrochemical gradient as do the electrons from NADH. So the electrons in NADH contribute more to generating this high energy proton gradient than do the electrons from FADH2, because these electrons drive 3 pumps, while these electrons are only going to drive this pump and this pump, only 2 pumps. So we've created this electrochemical gradient in the form of protons on one side of the inner mitochondrial membrane, held in by this by this outer membrane. So it's between the two membranes. So there's a lot of protons here, and there's not many protons here because they've all been getting moved from the matrix out this way. So to recap, energy in food has been transferred to the energy in NADH. So that was 2 different kinds of potential energy. Energy in food is a chemical energy, which is potential energy, stored energy. Energy in NADH is stored energy in the form of chemical energy. It's a kind of potential energy. And then that energy was converted to the energy in this electrochemical gradient. That's another kind of potential energy. It's not chemical energy because it's not in a molecule, but it's still potential energy. It's still stored energy. Okay, so we've just transferred we've transferred the energy from food to NADH to this electrochemical gradient. Now, this whole time that this has been happening, every time energy is transferred to a different form, every time it's converted to a different form, some of the energy gets lost as heat. And sometimes we say that it's wasted. The reason we say it's wasted as heat is because once it's heat, it's not easy to use that energy or convert it back into a different form. So that's often the end of the line for the energy transformations. So every time an energy transformation happens from this chemical to that chemical to a proton gradient, some of that energy is going to be quote unquote wasted as heat. It's not a true waste, right? Because a lot of organisms need that heat to stay alive. You know, if you're talking about higher order organisms, that heat energy is actually useful. We need to be warm bodied, right? The warmth in our bodies comes from this process. We don't have any other heat giving mechanisms. It comes from the burning of calories in the electron transport chain. Okay, so every time electrons are transferred, some heat is released and some is passed on to the next step.